everyone. I'm happy for this fantastic opportunity to tell you guys the story of how we tracked mitotic chromosomes using microscopy, super resolution, and deep learning. Now, let me start with saying a few words about the three-dimensional organization of chromatin. So in the past centuries, even, it's been a mystery how chromosomes are organized in space. And only recently, we begin to understand of this three-dimensional organization. And even now, the standard textbook view is that there's a difference in this 3D organization between the different phases of the cell cycle. So we know that in interphase, chromosomes are hardly discernible, while in mitosis, we can easily see them with light microscopy or fluorescent microscopy. And even between different stages of the cell cycle, there are obvious differences. We know that in metaphase, for example, chromosomes are compacted and they are aligned at the metaphase plate, while in prophase and prometaphase, they're longer and they are moving much faster and they are rearranging their positions. So it's really a very interesting question of how this process of chromosome rearrangement takes place, not just how they are arranged in space, but also over time. And there are several methods how traditionally people go about this problem. So we have fish where we have fluorescent probes, which are complementary to some loci on the DNA. And we hybridize these probes, which can allow us to see where these loci are located in space. We also have the whole chromosome fish on the bottom left, where this can give us information about the karyotype, the copy number of different chromosomes, any translocations that the cell has. Also, there's three-dimensional fish on the bottom, on the upper right corner, where we can preserve the shape of the nucleus in 3D, again fixing the cell, and see exactly where each chromosome territory is in 3D space. And of course, we have the high-C method and all its variations, which can give us even more detailed information about the structure of chromatin, the loop extrusion process, and so on. But an obvious drawback of all of these methods is that they're static. They're missing dynamic information about the chromatin dynamics, right? So with the fish, we can have a great picture of the karyotype, but we have to fix the cell, and we have no information about how chromosomes are moving over time relative to each other. The high-C, again, it may give us more detailed information about space, but again, it's a static method. And even it's complicated because we have to average many different cells. So even if we have information about the proximity between different loci or different chromosomes, these are average contacts. So we lose single cell information. Of course, there is single cell high-C as well, like this example that I have taken from this great paper on the bottom right, but it has problems of its own. So for example, it has so-called sparse coverage. We completely miss some of the contacts with single cell fish. And again, it's a static method. There is no information about the dynamics of the process. So to the rescue, we have microscopy, right? So with microscopy, we can see a living cell. Like what we do usually, we tag chromatin with some fluorescent markers, such as a GFP to H2B fusion, and we can take fantastic pictures of chromosomes using super resolution. But we have other problems here. We can't take super resolution images very often because this is damaging to the cell. You know, when we shine high laser power on the cell, this fluorescent marker is bleaching very fast. And also it's phototoxic to the cell, especially in this strictly regulated division when, you know, there's this thousands of different proteins and different pathways which have to be organized in a specific way so division can happen. And this has to do with the so-called pyramid of frustration, where we can have resolution or white exposure or imaging speed, but we have to choose, right? We can't have everything at the same time. So in order to do this, to get high resolution imaging of chromosomes, and at the same time do this very fast, we have to solve quite a few challenges, both in microscopy and in data analysis. So the first thing we have to keep in mind is that when we want to take a 3D picture of a cell, we have to think about how big the cell is. So for example, here we see a cell in, let's say it's in prophase when it's still flat. And we choose how many planes we need to do a 3D image. But later, when the cell enters prometaphase, if we keep the same settings, then it's going to bulge out of our field of view. So we lose that information. And even later, when it enters metaphase, it becomes rounder, like a sphere. So we lose even more information. So we have to either lose information or we have to keep adjusting our microscope settings every time the cell is changing shape. And this is not good because suppose we want to take images every second, which is essential for tracking chromosomes. Every second is important when we want to track chromosomes because they move so fast. We don't want to adjust settings every few seconds. So what we ended up doing is to anticipate the cell shape at the very beginning. So we calculated that we need approximately 22 microns. So instead of focusing on the beginning and end of the cell while it's still in prophase, what we ended up doing is to leave a bunch of empty planes. So we take 100 different Z planes, and most of them are empty at the beginning. But that's okay because they will fill up as time goes on when the cell starts changing its shape by entering prometaphase and metaphase. And this way, we don't have to adjust the settings every time. So we achieved imaging every 8 seconds. So every 8 seconds, we take such a huge 3D stack of 100 planes. So let's say if a mitotic cell is taking 40 minutes to divide, then this results in something between 200 and 300 3D images. And this is what such a movie looks like. Now here, every frame is a projection of a 3D image. And we can see that how fast we can image this cell. And of course, we can immediately see the next problem, right? So if we take images so fast and so often, then this is damaging to the cell. So we have to take care of the bleaching and the phototoxicity. And we found that we can somewhat alleviate these problems by lowering the oxygen. And this is a well-known effect because in cancer cells, especially, they grow in low oxygen conditions, the so-called hypoxia. So lowering the oxygen has a beneficial effect both on cell growth and also on it lowers the effect of bleaching. And this effect is not very noticeable at low laser power. So on the top two rows, we see that cells divide normally when we have low laser power, which is expected. But when we try to increase the laser to get a better image on the bottom row, we start getting defects in anaphase. So this phenotype can be rescued by lowering the oxygen. And of course, we're not the first to show this. I was just surprised how dramatic this effect is. So it really helps lowering the oxygen to 5% instead of 20%. And we can quantify this and see that the fraction of divided cells at any given time is always much better when they're grown and imaged to low oxygen conditions. On the bottom right, we see that this also has an effect on the bleaching. So low oxygen results in much less bleaching. But now we have other problems. So, okay, we can take images every 8 seconds, but how do we segment the chromosomes? How do we track them? So the first thing we did is we did denoising. You guys are probably familiar with the CARE network, content-aware image restoration. I'm sure many of you guys have already used it or have heard about it. It's really an amazing tool for restoring image quality. So it's a neural network which takes pairs of images. So it takes a low resolution image and a high resolution image, and it learns to predict what the image should look like, even if it's low resolution. So this now allows us to use low laser power, but still get a high super resolution image and not be phototoxic to the cell. So this is how we developed this pipeline, which we call FastChimp, which is a shorthand for facilitated segmentation and tracking of chromosomes in mitosis pipeline. So it combines three different neural networks. One is for denoising, like I just said. The other is doing segmentation, and the third one is doing registration. So registration helps with the tracking of transferring each segmentation mask from the current time point to the next. So on the top, we see our raw images. The middle row, the denoised ones by the CARE network. And then on the bottom left, we see the result of such a segmentation. Now, talking about segmentation, this is obviously a more difficult problem than usual, because what we're usually used to thinking about segmentation is just cells or nuclei, like we have on the left. And there are so many different tools already for segmentation of such so-called convex objects, right, which are round like a cell. But chromosomes, obviously, this is not the case for them because they have very twisted and complicated shape. So any standard method like watershed or even something more sophisticated like cell poles or other neural network usually fails. So we found this amazing tool called EmbedSeg, which is it was originally developed for other applications like C. elegans worms. But think about it, C. elegans look very much like chromosomes because they have a similar shape. So when I saw this paper, I decided to try it out, and it gave amazing results. So what we ended up doing is we first did manual segmentation of chromosomes for a single cell, which took us a few months. It's a very, very slow process. But once we accumulated enough data, we were able to train this network to do the segmentation automatically. But of course, even with automatic segmentation, there are still errors that need to be corrected later. So we had to develop this program that we call PyFix 3D that allows us to do manual corrections in time and space. Now, of course, there are many other programs which can do manual corrections, but most of them work on two-dimensional images or 2.5D. And this one allows us to rotate the view, select the specific area that we want to correct, and then give it the correct color. So this is a pre-recorded movie of such a process. So for example, we identified this pink chromosome that is supposed to be part of the purple round on the right. Then using something like a magic wand, we can draw around it and then fix it in a matter of seconds. So this is open source. You can see here the GitHub link. And this tool now allowed us to finish the process. So we do the segmentation, tracking, and then we fix the remaining errors manually. And this is what a complete segmentation looks like in 3D. But now that we have these segmentation masks for each time point, there's hundreds of them. So we are left with the final task of tracking. So we have to transfer the segmentation from the current time point to the next one. And for this, we use the so-called registration, which is simply deforming one of the images to match the next. So we have the so-called moving image, which is the one that needs to be deformed, and the fixed image, which is the reference that we need to match. And we see that if we take two time points of such images, even though they're only 8 seconds apart, they're already quite different. When we overlay them, we see that they're quite different. So first what we do is we do rough alignment, what's called a fine registration, which is simply moving the structure, translating it in space without any deformation of the chromosomes. And we see that the alignment is much better now, but still, because the chromosomes have actually moved a little, when we zoom in on the bottom row, we still see that there are some differences. So we do the next step, which is now non-rigid registration, which is deforming not just the whole structure, but also the individual parts, which are the chromosomes. And we see that progressively the alignment is getting better. And then on the bottom right, we see that the alignment is perfect. So this method allows us now to track the structure by this complicated looking graph, but actually it has a very simple idea. So we take the current segmentation masks, we transfer them to the next time point, and if they match, they match. So the process is complete. If they don't, if a specific chromosome doesn't match well, then we do an additional step, this here on H, where we do additional splitting of this chromosome. So one segment can match to one chromosome, another segment can match to another. And this is what the results will look like when we apply this pipeline on the whole mitotic cell. So here we have a HeLa cell with the histone H2B tagged with mcherry. Again, hundreds of 3D images. I'm only showing you 5. On the top, we have the raw signal after denoising, and on the bottom are the chromosomes which are already tracked. And for each of these time points, we can extract an image like a karyotype, where we can see each chromosome, its shape, and a bunch of other physical characteristics that I'm going to show you in a minute. So we were also interested in seeing which chromosome is which, right? Which is number 1, number 2, to do a real karyotype, which is not possible in a HeLa cell. So we also tried the RPE cell line, which is a diploid cell. So it has exactly 46 chromosomes. Almost all of them are normal. So the way we can do this is by doing some measurements on these chromosomes. So first we can measure the length and plot it on a graph on the bottom left. But we can also infer where the centromere is located just by looking at the shape. Because in prometaphase, and especially in anaphase, chromosomes are pulled away by the microtubules. So they're attached at the kinetochore. So if we see where the chromosome is most bent, we can infer the location of the centromere and plot it against the length. And now this 2D plot gives us complete information about the shape of the chromosomes relative to the karyotype, because each chromosome has a known length and centromere location. In this way, we can uniquely identify most of the chromosomes, except for those which are very similar. For example, 4 and 5 are very similar in shape, so we can't really tell them apart. What's also interesting here is that if you look at these 2 black dots on the bottom left, the ones labeled X and X prime, these are actually the 2 copies of the X chromosome, but they are not close to each other because one of them actually has a translocation and is longer than the other. This is a well-known translocation in the RP cell line, and it's amazing that we can pick it up with this pipeline just by looking at the shape of the chromosomes. It was also interesting to see how well these deep learning models that we trained are reusable for other cells, other experiments, even different microscopes. So we decided to try different combinations of either cell lines. So we use HeLa Kyoto, U2OS, or RP1, different tags, mcherry, EGFP, Stay Gold, which came out recently, and also different microscopes. So we originally trained the data on images from an area scan microscope, but we also tested a confocal one with different cameras and even different magnification. So as long as we scale the images to match the training data, we can still get results, quite excellent results, even with different microscopes, different tags and cell lines. And we can quantify this, and we can see that there is some deterioration in quality. For example, we see that the SNR improvement is getting lower the more we deviate from the original conditions. Same is true for the intersection over union, other metrics like the fraction of correct segmentation or the mean square error. But what's amazing is that we can still do this. We can still, with a little more effort, we can still do successful segmentation and tracking, even on these very different conditions. And this is just an example of 9 different cells, RPE cells, which were processed using this method. And we can see how powerful this is, because now we can look at the trajectory of each chromosome, of the change in its shape, even the compaction, decompaction over time. So this is just showing the karyotypes of these 9 different cells and the chromosomes that were identified. One interesting question that we wanted to ask is whether each chromosome, where is it in space? Because there was this debate in literature before, what happens to the homologous chromosomes, right? Because in a diploid karyotype, we have 2 copies of each chromosome. Are they always next to each other, or are they on the opposite side of the cell? Because there were arguments for both for both theories. So we decided to look at the chromosomes for the homologs and measure the distance between each of them. And we saw no statistical pattern here. So it turns out that in RPE cells, at least, there is no pairing or anti-pairing of homologous chromosomes, at least not in prophase. So they were completely indistinguishable from just random chance. We verified using FISH that our segmentation is correct, right? So we used a specific FISH probe for chromosome 2, and it matches exactly the one that we identified using the pipeline. We also did a different type of FISH, the multicolor FISH, which labels different chromosomes with different colors using a combination of colors, actually a unique combination of colors to label each chromosome. And again, there were good matches between the chromosomes that we identified by FISH and the ones that we identified using our method. Now, it was interesting to track what happens to the proximities of chromosomes. So what we did here was to artificially color code each side of the nucleus, either left or right or up or down, and see whether these 2 halves remain separate or they intermingle. And it turns out that they will either remain separate or they will mingle depending on the axis that we choose to separate the 2 halves. And this pattern was observed in all of the different cell lines. But to understand why this happens, we decided to look at each individual trajectory. So we represented each chromosome by its center of mass, and we recorded the trajectory of motion of each chromosome as it enters prometaphase and then metaphase and then anaphase. And then we clustered these trajectories using k-means clustering, using 4 clusters, and then we covered each cluster with different color, as we see here. So what immediately we can see here is that these clusters stay together. They don't intermingle, right? So the green one and the yellow one stay together. What was even more surprising was that when we average these trajectories and put an arrow at the direction of motion, we can see that the motion of the chromosomes is not random. It actually follows these well-defined vortices that we called mitotic flow field. So these red asterisks are actually the centers of these vortices. So the chromosomes on each side, we can predict their motion just by looking at their position. So the ones on the right and the ones on the left rotate in different direction once the cell enters prometaphase. And also what we did was to plot the distances between all the pairs of chromosomes at the beginning in prophase and at the end of telophase. And we see there's some correlation between them, but it's a weak correlation. So there are cases of chromosomes which, when they were initially close together, but later they moved away. And chromosomes which are initially distant, but ended up as neighbors later in anaphase. And we can even plot the positional correlation as a function of time. And we see that this correlation is getting weaker over time, which means that even though we have apparent conservation of the positions by looking at these clusters, inside the clusters, there is some intermingling. And also there is rearrangement as a whole by following this flow field, so that we end up with these exceptions. And we can even plot this as distance matrices. So here we have the pairwise distances between each chromosome and every other. And we see that this pattern at the beginning is roughly retained over time, but not perfectly. So there is some retainment of initial positions of chromosomes, but with many exceptions. And we see this pattern in all different cell lines that we looked for. Of course, there is great cell-to-cell variability. Sometimes we have 2 vortices, sometimes 3, but it all follows the same swirling motion. So to understand why this happens, we decided to take a look at the centrosomes. So here we tag the histone with, again, with M-cherry, but also the centrosomes with GFP. And we used light sheet microscopy to follow the same, both at the same time. And we saw that this chromosome motion that we observed is very well correlated with the motion of the centrosomes. So centrosomes are initially outside the nucleus, but after the breakdown of the nuclear envelope, they go inside. And exactly at that moment, we see this rearrangement of chromosomes. Here on the right, we can see the flow field with the arrows overlaid on the chromosomes and the centrosomes. So our working theory here is that it's actually the cytoplasm that's causing this motion, because cytoplasmic flow has been observed many times before, even in plants and different kinds of cells. Even there are simulations on growing microtubules which induce cytoplasmic flow. People have used different beads, and it's really a well-established process. So maybe we think that the motion of the centrosomes is causing this type of flow, and the chromosomes move according to that flow. So the motion is not really random, but it's not a direct one-to-one correspondence. It just follows the motion of the centrosomes. And that's a movie showing this process. So this flow field is obviously different at every moment. It depends on the current position of the centrosomes. That's why these arrows are changing over time. This one's one cell, now it's going to divide, and now there will be another cell. Also, it's important whether the centrosomes are on the same side or on different side of the nucleus, because in the first example, they were on the same side, but here we see that one is on the left, one is on the right. And this also has a very dramatic effect on the flow field, because we can clearly see now that the arrows are arranged differently, and this whole pattern is different. Talking about distances, we were also interested in looking at the distances not just between chromosomes, but between different loci on the same chromosome. So what we did was, for each chromosome, we fit a center line, again by using that software that I showed you before. And then for each pair of points on this line, we can measure the distance. And what's more, we can also measure the distance between each point on one chromosome and every other point on different chromosomes. So we end up with this distance matrix, which resembles the matrices that we have in a high-C experiment. So we have the chromosomes again on each axis, but it's not just one point now. It's all different loci on this chromosome, depending on our microscopy resolution, of course. And we can see, we can see both interchromosomal and interchromosomal distances in this way. But what we can do now is we can generate one such matrix every 8 seconds, and we can see how this pattern changes over time. So this movie now is showing that there is very little difference between frame to frame, because we see that the pattern roughly stays the same. But still, although nothing changes over a few seconds, if we compare the initial and the final matrix, there are quite a few differences. These are just some examples in the white rectangles here of chromosomes which were initially close together, but then they became distant or the other way around. So again, we have evidence of significant rearrangements of chromosome contacts before and after mitosis. And these are examples of individual chromosomes now zoomed, so we can also look at intrachromosomal distances. And what's interesting here is that if you look at the two homologous chromosomes, they're not folded the same. They're very different. And now imagine we do a high-C experiment and we decide to average these homologs. So we're going to get a picture which is not representative of either. It's just an average of chromosomes 1A and 1B, and so on. So it's very important to look at individual homologs, not just average them. Then we looked at the compaction of chromosomes over time. So we can do this several ways. We can measure the volume, the length, or also the fluorescence signal, because the chromosomes become not just smaller, but also brighter, because we have the same amount of fluorescent protein, which is crowded in a smaller volume. And these are all orthogonal methods of measuring chromosome compaction at very high temporal resolution every 8 seconds. And if we do this, we can observe some very interesting patterns. So the blue curve here on C is the volume and the yellow is the intensity. Of course, they're anti-correlated because, like we said, smaller volume corresponds to brighter chromosomes. So what we see initially is that there is condensation, obviously, in pro-metaphase. Then there is some leveling of this curve in metaphase. But then something very unexpected happens. There is dramatic change in the signal at anaphase. So it looks like there is brief decompaction in anaphase before the compaction resumes and becomes even more pronounced in telophase. And we can see this effect even at the level of individual chromosomes. We are fairly sure this is not a computational artifact, because we can see individual chromosomes and it's not a single observation. It takes a few minutes for this signal to drop and then recover. And we can even see it by looking at the individual sister chromatids, that there is some decompaction. And it's actually logical, right? Because you have the chromosomes still attached by cohesin rings before they are fully separated. And at the same time, they're being pulled away by the microtubules. So it only makes sense that they will slightly decompact at this time point, which was interesting because it wasn't observed before, at least not for human cells. I saw a paper about bacteria at some point, but it was very indirect evidence. No one had ever looked at microscopy of anaphase chromatids before in this manner. And it also makes sense because people have also used optical tweezers to extend chromosomes in vitro, and they're very pliable. It's very easy to extend a chromosome even several times its length. So I think this is a real effect, actually. We also looked at the condensation of one specific chromosome, but over many different cells, just to see if the condensation follows the same kinetics. And it does. This is an example of chromosome 2 in 9 different RPE cells. And we can see that the condensation kinetics between them agrees very well between replicates. So another interesting question we ask later is, can we compare between chromosomes of a mother cell and its daughter? So we image a single mitosis as before. We do the segmentation and tracking, but why stop there? Why don't we wait for the whole interface, follow the cell over time, and see what happens to the daughter cells 24 hours later or 20 hours later when they enter mitosis? We can do that, right? We can compare the location of individual chromosomes. Are they in the same place or are they arranged in a different way? So what we did here was to identify chromosomes 1, 2, and 3 in HeLa cells. So some of them have 3 copies. And look at the pattern of those chromosomes and compare between the mother cell and the daughter cells. And we can see that the two sister cells are very similar. If you compare the dots here, which represent on the right picture, they represent the center of mass of each chromosome. So we can see that, for example, the 3 red dots are on the same side. There is one blue dot close by, and then 2 other dots on the other side and the purple dot close by. But this arrangement is very different from the mother cell. So we can make the conclusion that there was significant rearrangement during mitosis, which we already showed with the flow field, right, with the distance matrices. But now we see it after comparing position across generations. So the mother cell is very different, but then the sister cells are very similar, which means that probably there wasn't much motion over interface. So once mitosis was complete, once the chromosomes found their territories, they stayed there. There wasn't significant rearrangement after this point. And we can see this in many different cells. Again, the mother cell is always different. The sister cells are similar. But sometimes we saw that this similarity in sister cells is only apparent when we rotate the nucleus. So initially it would seem that some of the sister cells are different. But after we artificially rotate the data, we see that they match exactly. So to see if there is such rotation, we went back in time because remember, we imaged the whole interface, not just mitosis in this experiment. So we went back in time and we saw here on the bottom that indeed for these cells, there was rotation for one of the sisters and not the other. And we computed again the flow field of this rotation, overlaid the arrows on top, and we saw that it's quite significant. So this can cause some apparent rearrangement of chromosome positions, but they're still arranged in a similar way after we compensate for this rotation. The final experiment that we did, one of the actually most interesting, was to look at chromosome bridges, because we already studied normal phenotypes, but what happens when there is a bridge, right? When there is some abnormal mitosis. So what we did here is we just use a stronger laser power to induce mitotic bridges. We applied the pipeline, segmented each chromosome, and then we identified which chromosomes form the bridges. So for example, in this cell, there were 5 bridges, which were both copies of chromosome 3, one of 6 or 7, and one of 4 and 5. And what we did was to compare the length of these chromosomes to a normal chromosome. And as expected, we can see that in anaphase on the bottom right, the yellow curve shows that, again, the chromosome is lengthening, But now this deformation is much, much bigger than the one we observed before during a normal anaphase. So this is more than 1 micron difference. And all of these methods are publicly available. One is called FastChimp. The other is PyFix3D. So they're on my GitHub account. You can guys see here the links. I can also share them in the chat in a minute. We also have the different cells available. So all of the cells that we analyzed, their raw data paired with the segmentation masks that anyone can use. They are in this Figshare repository. There is a link in the paper. So I imagine they can be used for many things. For example, anyone can train some other deep learning model, or they can look for patterns in the motion of chromosomes, trajectories, anything that we haven't thought about. And this is our group at our Sofia Euro-BioImaging node. Amazingly, we managed to fit almost everyone in 2 pictures, which is rarely the case. So thank you guys for your attention.